What? Uh, you were muted. What?
I said you're holding the fort for Team Bordeaux.
Yeah. I mean, they should be. Yeah, I don't know. They might
A
have a
lot of
meeting.
people today.
Great. Hey, Emmy. Here we go.
Hello.
Hello, hello. Okay, it's calm down now. So, welcome to Neuroccino on this Monday morning, 9:30-ish today. Today, Eva's going to present on skull, bone marrow, and depression. Looking forward to that. Take it away. Share the link in the chat, please.
Oh,
And then take it away.
1 second. Copy the link. Please. Um, okay, you can see it fine. Right, so I chose this paper because I don't know about you guys, but I had never really paid much attention to skull bone marrow. And I saw both of these papers were published in November. This one is in Nature and looks at aging, the relationship between skull marrow and aging in mice. And the one I'm going to talk about, it looks at depression in humans. But if you're interested about this other paper, you can go read it, or in the Nature podcast, there is an interview with one of the editors, actually, so you can get a shorter version maybe. But essentially, in this paper, they discuss how depression is in part an immune dysfunction, and there are elevated markers of both peripheral and central inflammation. So in both inside and outside the brain, we see inflammation markers. For instance, the more reliable ones are C2 reactive protein, interleukin, and tumor necrosis factor. And this is a bidirectional nature of the immune system. So both in immunotherapy, there are symptoms of depression, and also when people are administered anti-inflammatory medication, they also see an improvement in symptoms. So this is really linked together. And the way that people before have looked at neuroinflammation is through the mitochondrial translocator protein, which can be detected through, not detected directly, but we have a tracer that can be injected, and with PET imaging, we can see if this protein is being synthesized. So yeah, it's synthesized by a bunch of cells, and in depression specifically, it has been shown to be upregulated in the anterior cingulate cortex and prefrontal cortex. So essentially, we have brain inflammation, and there's also been shown to be just in general immune reaction, but these two are not really correlated. So we don't know how the two of these are communicating, or yeah, how they're influencing each other. So that's the point of this study, to look at other possible reasons, or not reasons, other possible mechanisms that could be mediating these two. So essentially, they take 51 depressed individuals and 20 controls. They take venous blood samples so that we can get an idea of the peripheral inflammation, and they have a range of peripheral inflammation for the depressed people. And then they also do a PET scan. With this PET scan, we can both extract the neuroinflammation as well as the activity of the bone marrow. So for that, we have to first segment the skull. And also here is how they segment the confluence of sinuses. So with this, we have several rows. First, for the skull, the facial bones, the occipital skull row, dorsolateral prefrontal cortex bit of skull, and then a parietal bit. So it's not the full extent because there's not really a delimitation. You cannot really say from here to here it is exactly facial and it's exactly parietal. So they just take a small row of each section. And they quantify the standardized uptake values for these, as well as the confluence of sinuses and the ACC, so that the ACC is taken as the neuroinflammation and the rest are the variables that they're looking into. So yeah, main message is we have periphery, we have neuroinflammation, and we have these extra-axial regions that maybe are mediating these other two that are not correlated. And so if we go into the results, I'll show you directly here. I grouped them mainly for results. So first of all, there is the brain inflammation together with the peripheral inflammation and the group explain the extra-axial variability. So the variability in the signal of these regions of interest. Then what we see in figure 4 is that the signal in the ACC, so the neuroinflammation, is correlated with the signal in the different rows after accounting for group and the peripheral inflammation. Especially in the confluence of sinuses, this was the strongest association. Similarly, for the peripheral inflammation and accounting for the other factors, we also have 3 instances in which we see a relationship. Finally, the only region of interest where they saw a difference by group was in the occipital skull. And here you can see how much of a difference and the distribution. This is after adjusting for neuroinflammation and peripheral inflammation. So yeah, the most interesting part I would say here is in the discussion that we're already in. So first of all, they found an association from the peripheral immune system to these different ROIs, and as well as the neuroinflammation to these different ROIs. There was no direct correlation between the central and the peripheral, and that therefore they think that mainly the confluence of sinuses, that was the most related one to the two of them, could be acting as a third party in this relationship, trying to facilitate crosstalk between the different immune mechanisms. And this aligns with previous studies that also didn't find a link between the central and the peripheral. And so, yeah, that is the first point. Venous blood pool is an important hub for central peripheral immune crosstalk. Then they say that for the facial bones and the parietal skull, they found weaker associations. And this could be due to the difference in the microscopic vascular channels. They have different properties across different bones. So this could be driving the results. But they point out that this is the first time that facial bones are taken into account in this kind of context. So I thought that was interesting, especially because the reason for it is that they are so close to the nasal passages, which have nasal microbiome. And so they are involved in immune function through this indirect link. And that was the reason why they included these facial bones. Lastly, the final main point is that they only found a relationship of group, an effect of group for the occipital skull. Since they were accounting for everything else that they measured, they say that there must be a mechanism that they're not measuring and they're not taking into account that is in this, that is affecting basically this difference that you can see in the graph before, because they were accounting for everything else. And that more research should be done into it. And yeah, they just conclude saying that there have been different patterns in, for instance, multiple sclerosis, stroke, Alzheimer's. So apparently skull bone marrow is being investigated for a lot of things, and I thought it was interesting. For the limitations, they just discuss the choice of the transporter, the choice of radio tracer, and whether circulating radiometabolites are contributing to the signal. But as you know, they say it's more or less not really a problem. But yeah, it's just what is most used in the literature at the moment anyways. So yeah, I hope you got the main message that this extra-axial area is or could be contributing to how some illnesses are or disorders are developed, or how they, there might be a way that we can alter treatments to target precisely this mechanism and understand more about this crosstalk between the different immune systems. So yeah, that was short, but yeah.
Thank you very much. So I know they applied this to depression in this case, but would this apply to any other disease, neurological or not, or inflammatory or not?
Yeah, I guess you could look into it in the same way. And the markers that they chose are also very widely used across different pathologies. But the crosstalk might be happening in more areas of the body, but they talk about how close areas are more likely to be influencing. So for instance, the occipital skull is more likely to be sending different molecules to occipital brain area. So the skull being so close to the brain is more likely to be doing this crosstalk, essentially. But perhaps for other disorders are more peripheral, it could be somewhere else, or...
Right, I'll give the floor to Eduardo.
Hi Eva, thank you so much for the presentation. The skull is so out of our comfort zone.
Yeah, definitely.
But it's nice to hear something about it. I have more of a question, but also a curiosity. So why to focus on the skull? Because I get it that it's close to the brain, but that would imply, I don't know, is there evidence that there's some, I don't know, like localized pockets of immune cells or complement proteins that live like maybe only in the skull marrows? Or I mean, I don't know, I'm not an expert on the immune system, so just to know.
Yeah, so it definitely has its role, and it's more likely to be localized, like I was saying. But from this other, I just listened to the interview of the other study, this one, and they actually identify specific cells in, I think it was cells in the bone marrow. So that are very important for aging, and that this serves as a reservoir. So
Like specifically in the skull.
yeah, specifically in the skull of mice.
That's always
So
we know everything about the mice. Not much about humans. Yeah, okay, that's really cool. I mean, like a brain immune system interaction is that's quite something. Thanks. I think Chris was
This was
after me.
next, yeah.
Yeah, thank you for the presentation. So yeah, as Eduardo said, this is like very far from what a lot of us study, but doesn't it show that there is of course in our domain we usually focus on like quick changes or like at first it's just in the brain cells themselves and mostly the neurons actually and we look at neurotransmitters, we look at brain structure and connections and stuff, but for some diseases and especially I mean depression, it's like a general state of, so first we always focus on the brain, but we forget that it's a brain in a body, and the body is very important. And the so with the inflammation thing, I've seen a bunch of studies also linking it to linking depression and a bunch of other diseases to the gut-brain axis. So isn't it that, of course, we are, as researchers, we are very focused on our domain, but it seems that for some diseases, we forget that there are other parts, but although also other scales, especially time scales. There was another paper, I think, that we discussed in Neuroccino about the
CSF.
communication between neurons like it was just a slow communication, like neurotransmitters were and other maybe it was not neurotransmitters but other chemicals, molecules that were just diffusing in the brain, so it could take hours, if not days, to reach the other side of the brain, and this could also link to this kind of studies, things that we always forget in neuroscience because we are well, in like study of the brain directly. Um, yeah, it's really interesting to see this kind of studies to remind us that it's not only the brain, right?
Yeah, definitely. I think that's also why I was interested in it, and especially to then learn that it had such a, yeah, such a localized, it's not very localized, but that there is more chance of a localized reaction. And I don't know, it's interesting how, yeah, everything is circulating, but then some patches are some things in some place. I don't know. It is good to take the more integrative view on what you were saying of the timeline. Definitely. Yeah.
Yeah.
Very good.
I not exactly a question, but a comment. Like I also keep seeing this studies about how like post-viral infection, like when you have a simple flu, for example, people can recover from flu, but they still have that post-viral depression, kind of like fatigue, and yeah, probably it's related to this like I don't know, like sinus infection. It's maybe so close to the brain, and then it infects affects the central inflammation or something. I don't know. Like maybe it's linked to that, but
Yeah,
I
I
keep
mean, it's
seeing
an
this
immune
like
reaction as it
post-viral.
is.
Yeah.
It's already a virus, so you have the peripheral immune reaction, and then it's sinuses, so maybe that's affecting microbiome, and then also you're just sick for days, so obviously you're gonna be fatigued afterwards.
Yeah. But they don't just call it the fatigue, but post-viral
Right.
depression. I keep seeing that terminology.
Yeah.
Uh,
No, I haven't
it
seen it
really,
before, but
yeah.
interesting. Yeah, so many ways in which we can be affected that we don't even think of. Yeah.
Yeah, thank you. It's very interesting.
That also had a question.
Hi, thanks Eva for this paper. I was thinking some women, I mean, at some point they get the menopause, and with menopause you get lower bone density, and so I guess also the bone marrow will be affected. So I was thinking, will be there any correlation? I mean, there is something connected with that. I think that when there is going to be these hormonal disruptions and stuff like that, then the risk increasing of some, I don't know, Alzheimer dementia or something like that. I'm not sure. But I was thinking if they talked about it, or you think that might be a nice study to see if there is more direct correlation with the lower density bone marrow and the increase of some diseases like that, because the immune system goes down and the proximity skull is losing the ability to fight whatever it is and flush out whatever we don't need to have in our brain. So
Yeah,
yeah, I
I
know
think
the menopause is not a disease, but it can lead to, unfortunately, to these things, evolutionary speaking, as well. You're not doing anymore your reproductive function, so you don't need anymore for evolution, sort of.
I definitely think it would be interesting. It's also a bit yeah, a bit more complicated because already the loss of bone density is a consequence, no? First, like you're having these hormonal disruptions, then there's the bone density, bone marrow, and then this would be this immune system. I don't know, in the scale of huge hormonal differences with the tiny immune, I don't know if it's tiny, but maybe the scale of this immune reaction would be harder to quantify or to see against the effects of having just very different hormone balance suddenly. But
Yeah.
for sure interesting. Like maybe maybe there's
I mean,
also
it could be interesting to see compare the correlations, like if there is one of those factors will be higher correlating than, like, I don't know, the level of estrogen, or probably, as you are saying, it might be the not as important, but yeah, can be
no, it's
interesting if
still
that's actually a thing, because about these neurodegenerative disorders is that there is something in our body that accumulates and we cannot flush it out. So immune system is not really fighting as it could. I don't know. Maybe.
yeah. I honestly, this is also very this is outside my comfort zone for sure.
Yeah. It's just discuss. Yeah.
Yeah,
Thanks.
but I thought it was interesting to, yeah, just bring attention to this, as Chris was saying, to just remind ourselves that the brain is in the body and, yeah, maybe pay a bit more attention to skull bone marrow in the future. Especially since I see papers coming out about this, like in the same month, these two, and yeah. So.
And I think it's fair to say we're all a bit out of our comfort zone here. So don't worry. Philip has a question.
Yeah, thank you, Eva, for the presentation. It was great. I'm just looking at the figure 6 and wondering because the depressed patients, they show like this bimodal distribution. So I'm wondering whether the authors also commented on that, or what can it mean? Whether like if the sample is maybe heterogeneous, or if the depressed patients can show different relationships.
I do remember reading some correction that they did to, but I don't remember which of the variables it was.
So they addressed it in some sense.
But I'm not sure if it was for this specific analysis they did the correction. Maybe it would be here.
Yeah, because in the end, I think the statistical analysis is done on like the averages of the mean.
Right.
So maybe they may be lumping like two
Mm-hmm.
different
They definitely
different
applied
groups.
some correction to one of the variables. I just cannot find it right now. Oh, but that was test of sphericity. So probably they
Okay.
did not account for that, and it was just, yeah, it was just looking at the mean. So missing part of the picture,
It was
probably.
just a curiosity.
You're pointing out, yeah. True.
Okay, thank you.
You're welcome.
So you started off with the Nature paper, and then you took us to the Brain paper, and you keep referring back to the Nature paper. So I was wondering if you could quickly speak to why you chose this one over the other and how they actually inform each other.
So if the skull bone marrow is out of my comfort zone, the mice skull marrow with the bio stainings is more outside of my comfort zone. So PET, I can understand. This was, yeah, more out there also in terms of the methods and stuff. But I don't know. I just heard it in the podcast, so that's why I'm a little bit informed, but not really in depth. And then I was just interested to see if we could see these kinds of things in humans already. So yeah, how they inform each other. One is more about aging and preserving this skull bone marrow. So it's actually more aligned with what Lilith was discussing of, yeah, how we age, our bones change, and then that also could be tapping into these reservoirs that then suddenly make us age even more. But yeah, I don't know if I can speak to how they inform each other or they're related, because I didn't actually read the Nature one. It's on my list.
No worries. Thank you very much. That's all the time we have this week. Next week, Marcella is going to present and Anna is going to host. So see you next Monday, 9:30. Have an exciting week in science. Bye.